All right, team, welcome to your pre-exam mission briefing. We're getting ready for Professor Pasquinelli's final, and today, our target is one of the big ones, RNA processing. Let's get you equipped to conquer it.
Okay, so first things first, let's size up our target, the final exam titan. You know, Professor Pasquinelli's questions are never just about memorizing facts, right? They're really designed to see if you can apply what you know when you're under pressure.
Yeah, a question like this can look pretty intimidating. I totally get it. There's a lot of info packed in there. But honestly, don't worry. Today, we're going to arm you with the right strategy to just break it down, piece by piece, so it's not so scary. So, our first piece of essential gear is all about understanding the 5' cap. Seriously, think of this as the helmet for your messenger RNA. It is the very first thing that goes on, and it is absolutely critical for its survival out there.
So what is it exactly? Well, it's basically a modified guanine nucleotide, and it gets added co-transcriptionally. And that just means it's put on while the mRNA is still being made. This isn't just some random decoration, it's a vital piece of equipment for the entire life of an mRNA molecule. Okay, this slide right here lays out the 3 critical functions you absolutely have to remember for the exam. The 5' cap provides protection from nasty enzymes that want to chew it up. It acts as an export signal, kind of like a passport to get out of the nucleus, and it's the flag that tells the translation machinery where to start making protein. So remember those 3: protection, export, translation.
And here we go. This is a classic Pasquinelli move. You know, she loves to test not just what things do, but what happens when they go wrong. So let's think about it. What if the capping enzyme is defective? What happens then?
Well, as you guys saw in your homework, the consequences are pretty disastrous. Without that complete cap, the mRNA is totally vulnerable to degradation. It gets trapped in the nucleus, and even if it does manage to escape, the ribosome just won't recognize it. The big takeaway is this: a failure in capping leads to a massive drop in gene expression.
All right, let's move down to the other end of the mRNA and see how we can build up our defense. You know, if the 5' cap is the helmet, then the poly-A tail is kind of like the shield protecting the rear. Now, a really key detail here is that this tail is not encoded in the DNA. It's added after transcription is already finished. And that process is called polyadenylation. That distinction is a super common topic on exams, so you want to lock that in.
You see, all these functions are totally interconnected, and the key player you really need to know is PABP. That's the poly-A binding protein. PABP latches onto that tail, protecting it from being chewed up. But it also reaches around and interacts with the 5' cap, forming this cool loop structure that makes translation incredibly efficient. So the cell's machinery doesn't just add this tail on a whim. It's looking for 2 very specific signals in the pre-mRNA sequence. First, there's this highly conserved AU-AUAA signal. And then a little further downstream, there's a G/T or a G/U rich element. It has to see both of those to know exactly where to cut the mRNA and start adding all those A's.
And this slide, it just brilliantly illustrates how important that specificity is. Like you explored in Homework 5, what if the cell's polymerase accidentally added a bunch of U's instead of A's? Well, the whole system just collapses. PABP cannot bind to poly-U tail, and without PABP you get 0 stability, no efficient export, and no translation loop. The end result? Gene expression just plummets.
Okay, armor is equipped. Now let's take what we know and apply it to some real exam-style questions. It's time to see how our defenses are going to hold up in a little combat simulation.
First up is a really common scenario. You get some data, and you're asked why an mRNA might be unstable. The moment, and I mean the second, you see unstable mRNA, your brain should immediately jump to the 3 pillars of processing: capping, splicing, and polyetylation. A failure in any one of those could be the culprit.
So how do we solve this thing? Well, the key is just to evaluate each option against those fundamental rules of RNA processing we just went over. Let's analyze the possible answers from that pre-final question and see why they work.
And this just shows so perfectly how different failures can lead to the exact same outcome. If the mRNA isn't polyadenylated, it's going to be unstable. If an intron is retained, well, that intron probably has a premature stop codon in it, which flags the mRNA for destruction through a pathway called nonsense-mediated decay, or NMD. So you see, multiple answers can be correct because multiple types of errors all lead to instability.
Okay, let's switch gears to another classic question type, predicting the result of a specific mutation. This really tests your ability to trace the consequences of one little error all the way through the entire biological process step by step. Hey, remember that GT-rich element we just talked about? The second signal for polyadenylation? Okay, using the same logic from homework 5, let's trace the consequences of mutating it. What do you think is the first domino to fall?
So the machinery sees that first signal, the AAUA, but without the downstream G/T signal, it can't cut. So what does RNA polymerase do? It just keeps on chugging along the DNA, transcribing away until, eventually, it hits another kind of hidden poly-A signal way further downstream. The key thing here is that the process doesn't just stop, it finds an alternative. But that leads to an altered mRNA with a much, much longer 3' UTR, which can totally mess up its stability and translation.
And that concludes our combat simulation. Let's head to the mission debrief for your key takeaways. You're going to want to lock these points in your brain before you deploy for the exam.
All right, these are your core tactical points. The cap and tail are armor. Their 3 functions are clear. Signal mutations don't cause a full stop. They cause a change. And pretty much any defect in processing leads to an unstable mRNA. So if you're ever stuck, just take a deep breath and trace the pathway all the way from the beginning.
And you know, the really crucial thing is that this method, breaking down a process, understanding its key signals, and then predicting what happens when things fail, it isn't just for RNA processing. You can apply this exact same strategic approach to transcription, translation, and replication. You are totally ready for this. Good luck.